Yes, for the young scientist session, we have two very talented students in biochemistry from the University of Geneva. And they are performing a 1-year internship of master thesis in ethics. The first speaker is Florian Shala, and the second is Sendia Ferreira Lopez. Donc, maintenant, now we give the floor to Florian.
Yes, thank you, Sam. So good afternoon, everyone. I'm very happy to be there and to present to you our results. So I'm Florian and I'm doing one internship here in epithelics for my master thesis. I'm trying to develop new immunocompetent model based on previous work done by my colleague Gao Gunazinam and Guy Barba. And the next few slides will show some results that we've got. So in epithelics, we developed two in vitro models representing the upper and the lower respiratory tract. So the first model is the muscular one, which has nasal, tracheal, or bronchial origin, and which is also available with human fibroblast. And our second model is a smaller model, which has smaller airway origin, and which is available with human fibroblast. So to mimic what is happening in vivo, some immune cells should be added to these models. So this is the aim of my master thesis, where I try to develop a new muscular model in co-culture with dendritic cells. So dendritic cells originate from hematopoietic stem cells in the bone marrow. These hematopoietic stem cells can differentiate into granulocyte macrophage progenitor or in dendritic cells precursors. These dendritic cells precursors can further differentiate to plasmacytoid or in classical dendritic cells. So these cells issued from the dendritic cells precursor are present in the lungs when there is no inflammation. In contrast, the cells issued from the granulocyte macrophage precursors are found in the lungs when there is an inflammation. In our model, we focused on the second group because it is easy to isolate monocytes from buffy coats and differentiate them into monocyte-derived dendritic cells. So some previous paper affirms that there is a gradient density of dendritic cells going down the respiratory tract. So that's why we use the muscular model as a template for our model, because it is more representative to what is happening in vivo. Furthermore, the dendritic cells are found under the epithelium, and they can sense some pathogen. So that's why we put our dendritic cells in the basolateral part of our insert. So to prepare our dendritic cells, we isolated PBMCs from buffy coat by fecal gradient. Once we've got the PBMCs, we put them in a flask and let them adhere 1.5 hours to have the monocytes. We wash it 3 times to remove all the cells we are not interested in. Once we've got the monocytes, we differentiate them during 6 days using a cocktail of cytokines to obtain immature monocyte-derived dendritic cells. To do the maturation, we put the cells in presence of LPS during 2 days. So first, we wanted to characterize our monocyte-derived dendritic cells. So to do it, we've done some immunofluorescence. So here I show some results for immature dendritic cells and mature dendritic cells. And as you can see, some markers differs, the expression of some markers differs between immature dendritic cells and mature dendritic cells. By the battery of markers that we've tested, we conclude that our immature dendritic cells are positive for these markers and negative for this one. So after that, we wanted to know if our immature dendritic cells can phagocyte, as mentioned in previous paper. So to do so, we've done a phagocytic assay using pH-Photo red zymosan. So these are bioparticulates conjugated with a dye, which become fluorescent as the pH become acidic. In the cells, the acidic compartment are found in the lysosome, and as you can see, we have a strong signal suggesting that our cells are able to phagocyte. So to confirm these results, we have done the same experiment, but this time using diesel particulate. So as you can see, 1 day and 2 days post stimulation, the dendritic cells are able to phagocyte. To construct our model, we use the immature dendritic cells that we put on the basolateral part of the insert during 1.5 hours to create our model for experimentation. So to confirm that our dendritic cells are present on the basolateral surface of the insert, we've done a cross-section of it. So as you can see, we have two distinct cell population, one which is the fibroblast and the second one which are the dendritic cells. So to confirm that our second cell population are dendritic cells, we've done some electronic microscopy. And here we have the confirmation that the dendritic cells are present in our mucillar with human fibroblast model. So knowing that our dendritic cells are present in our model, we wanted to characterize them and to know how long they are in this model. So to do so, we've put a live dye called cell tracker on our dendritic cells to observe them in co-culture. And after that, we've done some immunofluorescence using the CD86 marker with time. So as you can see, the cell number remains stable in time, while the CD86 signal decreases with time. So with that, we've concluded that our dendritic cells are present in our co-culture until 7 days, and that the CD86 signal decreases from day 3. So knowing that our dendritic cells are present from day 1 to day 3, we wanted to do some stimulation to know if our system could respond to any pathogen. So to do so, we applied some LPS on the apical surface of the insert. And here are some results. So we had 4 different conditions. The blue ones, what I will call monoculture, are mucillar with human fibroblasts with or without stimulation, and the orange one are the co-culture with dendritic cells with or without stimulation. So as you can see, the adhesion of dendritic cells does not affect the transepithelial resistance and the cilia beating. So to complete our results, we wanted to know if other stimuli could have an effect on the secretion of different cytokine. So here we use the poly-IC, which mimic the viral infection, house dust mite, and diesel particle. And as I said, we look at different cytokine. So here are some preliminary results that we've got. So these results are expressed as percentage of in untreated, sorry. And here we have an illustration that our model is more sensitive than the monoculture. So as you can see, the monoculture, we have a 2-fold increase for when the dendritic cells are present in the detection of interleukin-8 and interleukin-6. In contrast, our co-culture model, when we applied some poly-IC, is less sensitive than the monoculture. Here we have a 5-fold increase of interleukin-6, and we have a 10-fold increase for the monoculture. So knowing that our model could respond to a stimulus, we wanted to know if our dendritic cells were able to phagocyte in co-culture. To do so, we have done once again a phagocytic assay, and as you can see, we have some signal which suggest that we have that our cells are able to phagocyte. So in conclusion, our dendritic cells mucillar model can respond to stimuli and may also have different response to it. And our dendritic cells are able to phagocyte. So our second immunocompetent model is based on pulmonary macrophages. So we are able to extract macrophages from lungs, froze them, and thaw them. So once we thaw them, we wanted to characterize it by flow cytometry. So to do it, we use the DRAG7 to separate living cells from dead cells. And we use the CD45 markers to separate immune cells from other cell type. In another sorting that we've done with CD68, we showed that we noticed that there are two population, one in majority which is CD68 negative and one which is CD68 positive. With that, we wanted to characterize our macrophages. So we used a panel of different markers, and we concluded that our macrophages have an alveolar type and that they are all positive for CD11b to CD206 and negative for CD14. Furthermore, a small population is positive for CD68. To construct our model, we just thought some macrophages that we isolated, and we basically distribute them on the apical surface of a small layer with human fibroblast model. So here we put some cell tracker to show that we have a good viability. We have a good viability for our macrophages. And here we've done a phagocytic assay to show that our macrophages are able to phagocyte in co-culture. Finally, we transfer our model to VIRPath for electronic microscopy. So here we look 24-hour post-adhesion and 72 hours post-adhesion of the macrophages. So as you can see, 24-hour post-adhesion, the macrophage seems to be on top of the cilia, while 72 hours after the adhesion, the macrophages are in close contact with the epithelium. So in summary, our monocyte-derived dendritic cells are characterized or positive for this marker and can be frozen. Our co-culture model is stable up to 7 days and can respond to different stimuli. The macrophages isolated from lungs are homogeneous and characterized as alveolar type and also can be frozen. And our macrophage smaller with human fibroblast model is stable up to 7 days. In Epithelius, we are looking forward to construct a new model with macrophages and in co-culture with alveolar. So today we are able to do dendritic cells mucillar with human fibroblast and the macrophages smaller with human fibroblast model. And in the future, we're looking forward to construct the dendritic cells smaller with human fibroblast and the macrophages mucillar with human fibroblast model. So thank you all for your attention.
Thanks a lot, Florian, for your very nice talk. I have to say it is your first talk, and congratulations. So you have a large list of questions. I'm sorry to pick up the two first one, and you would answer later on directly. So the first one is that are you stimulating concerning the DC mucillar DC model? Are you stimulating from the basal side or the apical side of the insert? And on what side are you sampling for cytokines?
Okay. So I apically applied we've done the stimulation on the apical surface of the insert, and we just collected the cytokines in the basolateral. We collected the cytokines in the medium where the inserts are. Okay.
Last question, and we will need to switch to Cynthia. Is what happens if the fibroblasts are absent?
Well, this is an ongoing project, so we're looking forward to compare what happened when there is a human fibroblast and when there is no human fibroblast. But this is an ongoing project, and I'm actually working on it.
Perfect, thanks a lot. I think we need to move to Cynthia's talk now on another topic on alveolar models. Thank you very, very much, Florian.